At this point in time, we already learned that individuals other than identical twins have different inherited traits because the DNA of each person is not exactly the same as that of another person. Examples of inherited traits in humans include eye, skin, and hair color. Sometimes, diseases result from an abnormal DNA sequence in a parent being passed to the next generation. Hence, any permanent change in the primary structure of the sequence of nucleotide residues in DNA is called mutation. A mutation can also be defined as an error in the base sequence in a gene that is reproduced during DNA replication. Such errors alter the genetic information that is passed on during transcription. The altered information can cause changes in the amino acid sequence during protein synthesis. Sometimes, such changes have a profound effect on an organism. If the mutated gene results in a partially or completely non-functional protein that is important for an organism to function, the health of the individual may be diminished. Some mutations may also result in premature death. A mutagen is a substance or agent that causes a change in the structure of a gene. Radiation and chemical agents are two important types of mutagens. Radiation in the form of ultraviolet light, X-rays, radioactivity, and cosmic rays has the potential to be mutagenic. Ultraviolet light from the sun is the radiation that causes sunburn and can induce changes in the DNA of the skin cells. Sustained exposure to ultraviolet light can lead to serious problems such as skin cancer. Chemical agents can also have mutagenic effects. Nitrous acid is a mutagen that causes the amination of heterocyclic nitrogen bases. For example, nitrous acid can convert cytosine to uracil. Mutations that occur in multicellular organisms can be categorized as either somatic mutations or germline mutations. Somatic mutations, also called as acquired mutations, are mutations that occur within cell types that are not involved in the reproduction or somatic cells. When a mutation arises in a somatic cell, then the mutation is limited to that cell or any of its cell division descendants within the organism. Cancer is a disease that results from somatic mutations in genes that are responsible for cell growth or cell differentiation. Such mutations causes the formation of a mass of mutated cells or cancer cells called tumor. For example, a skin cell can acquire a mutation very often from exposure to UV radiation, which causes it to begin to rapidly divide or replicate and form a tumor. Germline mutations, on the other hand, are mutations that occur within germline cells. Germline cells are the cells that are involved in reproduction, such as sperm and ovarian cells. If a mutated germline cell is passed on to an offspring, then it results in a constitutional mutation, which is a mutation occurring in the nuclei of every cell of the offspring. When a constitutional mutation causes a negative health condition, it is then referred to as genetic disease. Mutations can also be classified as a small scale or large scale, depending on the number of nucleotides involved in the mutation. Small-scale mutations involve a change in a small number of nucleotides, usually 1 to 3 nucleotides within a single gene. This type of mutation is subdivided into 3, and these are point, insertions, and deletions. For point, it is a single incorrect nucleotide that takes the place of the original nucleotide. This often happens during DNA replication. For insertions, it has one or more extra nucleotides that are inserted between two of the original nucleotides in a gene. Lastly, deletions includes a single nucleotide or a short sequence, usually 2 to 3 nucleotides, that are removed from a gene. Large-scale mutations, sometimes called as chromosomal mutations, involve changes in large sections of chromosomes. It is also subdivided into three: deletions, duplication, and inversion. For deletion, a large section of DNA within a single chromosome is removed or deleted. For duplication, a large section of DNA within a single chromosome is duplicated and reinserted. Lastly, for inversion, a large section of DNA within a single chromosome is inverted. The inheritance of one or more mutated genes can result in a genetic disease. Normally, individuals have 2 copies of each gene in the nucleus of every somatic cell in their bodies. A monogenic disease then occurs when one or both copies of a particular gene contains a harmful mutation. Monogenic disease is subdivided into two: the dominant diseases and recessive diseases. Dominant diseases are monogenic diseases that occurs when both copies of a gene are mutated. An example of this is sickle cell disease. For recessive diseases, these are monogenic diseases that occur when one copy of the gene is not mutated and one copy is mutated. An example of a recessive disease is cystic fibrosis. Lastly, a polygenic disease occurs when one or both copies of multiple different genes contain a harmful mutation. Example diseases for these are hypertension, coronary heart disease, and diabetes. And that ends my presentation about mutations. Thank you for listening.